Yes, good evening to all the participants. Welcome to the day 12 session on B cell epitope design in internship and certificate course in AI and ML approaches of drug and vaccine design. Before going to today's session topics, can we recap the last session topics? So, what exactly did we discuss in the last session? Very good. We discussed on T cell epitope based vaccine design. And what else did we discuss? Yeah, participants can come with your answers. What exactly did we discuss in the T cell epitope based vaccine design? Yes, we discussed majorly on a public domain database called Immune Epitope Database. In this Immune Epitope Database, we have plenty of tools and databases. And if you see in this IEDB database, I directly take you to the database and start explaining. So for going to the IEDB, iedb.org is the URL. So inside that, we can able to search the epitopes for a specific diseases. Not only that, we can able to search for specific host. We can also search epitopes for specifically for MHC1 alone or MHC2 alone or for both MHC1 and MHC2. And we can also find the epitopes from T cell based assays, B cell based assays. Okay, and also we can predict any type of epitopes. We have subtypes for T cell, we have subtypes for B cell, we can also search for that. So, whatever experiments done in wet lab, so and the peptide generated, so these data are present in database. And if you see, peptide epitopes are around 17 lakhs. Non-peptide epitopes are 3,316. Like this, you can see this population. Yesterday, basically, we worked on the MHC1 specific T cell epitope prediction and MHC2. So these are the two things we have discussed on yesterday. Okay, and if you see, we also have discussed that we can predict immunogenicity, processing, binding, elution, etc. So for that, I have shown it separate URLs. For predicting toxicity, what is the website we have gone? Very good. We also discussed something on docking, simulation, etc. Isn't it? Not just screening the epitopes, but also we have discussed like that. So just now I asked you one question. What is that? I was mentioning you on what is the website? Very good. The answer is toxin pred. That is the exact answer. Okay. So toxin pred server is used to predict toxicity of peptides. And if you want to predict the toxicity of small compounds, small molecule compounds, use Protox 3. But whatever we are discussing from day before yesterday is related to peptide-based vaccine design. So for predicting specifically for peptides, we have to go through this toxin pred. Okay, and similarly, we have seen immunogenicity tool is also available in epitope in this IEDB database, and we have also seen other than IEDB, we have also seen one more major web resource. Do you remember that? What is that? Raghava web server. Do you remember? So this scientist from I Square TD has has developed lot of tools and databases for working on immunoinformatics. Okay, so these are the things majorly we have discussed. And can I ask you a few questions related to this? So, so whatever epitopes we predict, it can be TH, that is high helper T cell epitopes, or cytotoxic T cell epitopes from a particular virus or a cancer antigen. This we have to do molecular docking, isn't it? So to which molecule we can dock? Whatever T cell epitope predictions we have done, it can be TH or TC. After predicting and after screening the epitopes, okay, you'll get one best epitope, isn't it? That best epitope you try to dock with one protein. I have explained you about the docking and simulation on yesterday. So what is the molecule, protein molecule? Not exactly I'm asking this. I'm asking you like if the peptide is obtained from a virus source, like a Nipah virus W protein source, for example, what is the macromolecule we can choose for docking? What is the major important target we discussed on yesterday? Of course, whatever it can be a virus or cancer antigen. SVC students can answer. If only if I tell SVC students to answer, I think they can answer. If you know the answer, SVC students can also answer. So whatever the peptides we predict from the Nipah virus W protein source yesterday, I told you we have to talk with alleles. What allele protein? Human allele protein. You remember? What is that? What protein distinguishes? What set of proteins distinguishes even siblings? There is something called phenotyping. What molecule phenotyping? See, I have given. So, yes, very good. The answer is HLA, human leukocyte antigen. I told we have specifically taken a population to work, isn't it? Which population I tried on yesterday for predicting TH and TC epitopes? You remember? I specifically taken one population. Which community or which state population I've taken? Domestic horse? Oh no, actually, we have taken Keralites, isn't it? We have I just taken yes, good. So I've taken Nipah virus W protein, and then I've taken the Kerala community people. You have to remember up to that. Okay. However, yes, Kerala Malayalam is exact. So this is what we have taken that alleles. We have searched specifically what are the HLA alleles or HL, and then we have also checked for HLA that DPDQ that is for MSC2 alleles. For MSC2 alleles, we don't have specific alleles for from Keralites. So I have taken to UP and then I have we have predicted the MSC2. So whatever that HLA ABC or HLA DPDQDR we have, this protein sequences we can get from even NCBA GenPept. From NCBA GenPept, we can take that allele protein and then we have to model it using Swiss modeler. And whatever you have is the primary target. And whatever sequence of steps we make to screen the epitopes, that is a ligand actually. You screen the best ligand, that is the best epitope, and try to dock finally with that MHC molecules. Okay. So if you're not doing docking and simulation, whatever you have screened the epitopes is in vain. We should utilize it, isn't it? Now let me go ahead with today's session, B cell epitope design. So today we'll be discussing on many concepts, especially we discuss with the B cell epitope biology, prediction tools on IEDB, and we have two types of epitopes in B cell. One is linear sequence, the other is discontinuous 3D structure based epitope. Okay, I told you this is more precise, discontinuous 3D structure. So again, we can discuss we recap the topics. Computational antibody design in general, an antigen and antibody structure modeling, docking, vaccine databases tools. So this is what we'll be discussing it on today. So first we discuss about the Raghava web server. We have two types of B cell epitopes. One is linear, the other is conformational, isn't it? So about this, we have different servers in the Raghava web server. We can try to predict the specific epitopes based on machine learning technique. So ABC Pred is basically we work with the artificial neural networks. CB Top basically works with uh, SVM, support vector machine. Like this, we have different types of epitope prediction in this URL mentioned below. Okay, and also these kind of URLs I've also pasted in the WhatsApp web. Yeah. So you can also look through that kind of a tools. And the next is innate immunity and miscellaneous servers. Okay, so specifically if you want to design any specific vaccine, can be a DNA based vaccine or immunomodulatory RNA based vaccine, or if you want to bring a microbial polysaccharide vaccine, the vaccine can be made from any of these biomolecules, isn't it, other than the live and attenuated for kind of viral vaccines. So whatever types of vaccines we like to discover, including this vaccine adjuvants, I was mentioning you that even I have developed a kind of a tool for vaccine adjuvants. Similarly, he has, but this is basically he has developed for immunomodulatory DNA. This is something different from what I have done. So I have done with the plant-based vaccine adjuvants, but he has done related to this. Like this, so many tools are there for vaccine as well as vaccine adjuvants. IEDB. This is what we are discussing from day before yesterday. In IEDB, not only we can predict the T cell epitope, we also have a specific tool for B cell epitope prediction. Okay. And related to B cell epitope, just a recap, we have two types of epitopes in B cell. One is continuous residues or linear epitope. The second is discontinuous residues because you see, you have equal space in these residues from 19 to 23. But here we have irregular lengths. The length is irregular. Okay, this is smaller length, this is larger length, and this is very smaller length. So this why we have this kind of change in length in the amino acid residues. The reason behind is here we just take the sequence for checking the binding, but here we take the structure of the epitope. So after knowing the sequence, we have to predict the structure. For predicting the structure of epitopes, we already discussed in previous sessions. We have a server called pepfold3. If you put the sequence there, you can get the structure. If you get the structure, the affinity of the antigen antibody is more precise if you have the structures. So that even the amino acid, whatever present in the epitopic, that is antigen epitope, is going to bind inside the paratopic region of the antibody. Okay, so that's what we discussed. Like two types of B cell epitopes are available, and T cell receptor we already discussed on yesterday. B cell receptor, which is most important for this B cell immunity, humoral immunity. Okay, so the structure is slightly different with respect to the T cell receptor. It looks like an antibody like a structure, isn't it? And the top one, whatever we see, is the B cell receptor recognition. Okay, and we can see the 3D structures of antibody antigen complexes. We can also take the sample files from the PDB website. That's what I showed. We will be seeing the same in the subsequent slides. When to use epitope prediction methods? When verified thoroughly that no information is available in the EDB on the antigen of your interest. See, we already have deposited epitopes in the IADB. Then why we have to go this kind of a methods like going with the T cell epitope prediction methods or B cell epitope prediction methods? So this is what I'm giving you. Determine the candidate antigenic determinants in an antigen of our interest other than epitopes provided in the IEDB. So for this purpose only, we are using the T cell epitope and B cell epitope tools. Now, related to if you want to check the affinity of antigen and antibody, the best example you can also download from PDB is the PDB ID is 1E08, no, not 0, 1E08. This stands for immunoinfluence virus hemagglutinin complex with neutralizing antibody. You know, this hemagglutinin is the target protein of influenza virus, and the antibody is from the human source. So if a person is infected from flu, okay, then obviously you can find this kind of interaction. This antigen is binding with our human antibody. So can we see this structure specifically in Discovery Studio Visualizer? In the visualizer, I just segregated this. There is in hemagglutinin, we have chain A and chain B. Okay, and we have this antibody. The antibody, how it looks like, antibody is like a Y-shaped arm, isn't it? This is how we can see a Y-shaped arm. Okay, antibody is like this. And whatever regions present in the antibody, I told you these are paratopes. And whatever binding to the antigen part which is binding to the antibody is what we call it as epitope. Okay, you understand here the epitope, the B cell epitopes, how it looks like. And so this is from human source, obviously, and this is from the virus. Okay, so like this, we can able to see the structure. And now we can see natives. If you want to, whatever PDB structures you can get, you can also segregate these two proteins. You can segregate this as protein 1 and protein 2, and you can do protein-protein docking through protein-protein docking servers. After docking, you can check the energy also. Even that kind of this you can use this as a sample file for docking protein-protein molecules. Now let me go ahead with another thing, another example, a PDB ID 1VXG, where we take the native sperm whale myoglobin. Here, whatever I discussed, the second type of B cell epitope, that is discontinuous. So if the epitope is binding, if you know the 3D structure of the epitope, then the 3D structure of the epitope can bind inside the protein also, isn't it? Few residues are binding inside. So you can able to see in this example, the epitope is binding with almost showing so many hydrogen bonds with the protein. Okay, some sites are buried inside the protein. So the example is this native sperm whale myoglobin. The structure itself, whatever protein you download, it comes along with the peptide. You got it. So I didn't just dock a peptide. So already in the structure, you can find peptide-based protein. So if you just download this PDB ID, you can visualize everything. Okay, something about B cell epitope prediction. In IEDP also, we have exclusive tools to work with it. First, we discuss on the algorithms we use for the types of B cell epitope prediction. The first type of epitope is the linear epitope prediction. For this, we can use machine learning algorithms. Both positive and negative training data sets are used. And if you see the examples we can take is random forest. So there is one B epitope prediction. We call it as PepiPred 2.0. We also have PepiPred alone. That is whatever PepiPred initially mentioned is 1.0. Yeah. We also have specifically a version called 2.0 version. Okay, so both works under random forest machine learning technique. And the second thing is artificial neural network. Using this, they have implemented ABC-PRED program. And similarly, SVM, BC-PRED, FBC-PRED. Okay, for linear epitope prediction, we can use this machine learning techniques for predicting the epitopes. And if you see the sequence-based epitope prediction, that is what we discussed as linear epitope, uh, specifically in IEDB, okay, specifically in IEDB web server, we can able to predict through multiple methods, especially all this uses this amino acid physicochemical properties for estimation of for identification of linear epitopes. Okay, first let me go ahead with the types of methods in linear epitope prediction, specifically in IEDB. So whatever I am telling, linear epitope is based on sequence based epitope prediction. Number one is futures which have been correlated with the location of continuous epitopes. Number two is the beta turns. So using the beta turns, the Chow and Fastman has proposed a method to predict the epitopic peptides. Like for example, if you feed Nipah virus W protein, whatever we have used as an input for the previous session, here also I can input the same sequence as an input, and whatever epitopes we can generate around 1000 epitopes are based on beta turns. This was proposed by Chow and Fastman. Okay, and similarly, you can see surface accessibility phenomena. We can also predict the epitopes. This was proposed by Emini. All these tools, whatever I am showing, are available in IEDB. The next is based on flexibility of amino acid residues, proposed by Corpus and Skulls. And the next is based on antigenic propensity, that is occurrence of residues in epitopes, proposed by Kolaskar and Tongoyankar. And the next is the hydrophilicity, based on the hydrophilicity of the epitopes by Parker. Now, this is the second method of epitope prediction, that is based on the 3D structure. This we also call it as discontinuous epitope prediction. Here also we have different algorithms to predict the epitopes. Number one is based on the geometrical properties, which combines with amino acid scales. The examples of the programs are Discotope, ELIPRO, and CEP. And based on geometric properties and amino acid scales. This is a second type. For this, we can use EPSVR. And we can also use protein-protein docking algorithms to check the protein docking affinity, that is antigen antibody affinity. Now, let me go ahead with the linear sequence epitope prediction. Number one is the Chowan-Fasman. As I earlier mentioned, the epitopes are predicted based on the beta turns. So, what do you understand by the beta turns? One should remember the major secondary structure element present in most of the protein is nothing but alpha helix, which accounts for more than 52 percentage of the secondary structures in various proteins or alpha helices. Next to that, you can find the beta sheets, and then only you can find uh, loops, turns, coils, etc. Isn't it? So generally, I told you this proline and glycine won't contribute to the alpha helices. So it can contribute beta sheets, obviously, isn't it? So we have a kind of a propensity scale. Okay, we have a propensity scale, and if you see this glycine and proline, these are helix breakers. We call these amino acids as helix breakers. But we also have few amino acids which form the alpha helix. Like if these amino acids, alanine, glutamate, leucine, okay, and methionine, okay. So glutamate, yes, sir. So whatever we have, okay, these amino acids can form the glutamate is not G. What is that? Glutamate, Q. So G for glycine, okay. So Q for glutamate, and glutamic acid E, isn't it? Glutamic acid we use for E. Okay, so whatever amino acids we take, okay, so these amino acids form the alpha helices. Okay, these form the alpha helices, but these are the alpha helix breakers. Now let me go ahead with the proline C. For the proline, we can see the value is higher. And similarly for the glycine, if you see the value is higher. When compared to the other amino acid values, these values are higher. Similarly for asparagine, the value is higher. Okay, so these, if you find the values are higher, these are contributing to the alpha helices, and these are contributing to the beta sheets. And whatever values you can find lesser, these can contribute to the alpha helices. But in our case, we predict based on the beta turns. You got it? So lower the value, the higher the beta sheet formation. The higher the value, am I right? No. Higher the value, it forms the beta sheet. The lower the value, it forms the alpha helix. Got it? So this proline is not going to form alpha helix, but it can form beta sheets or beta turns. This is how we can able to understand it through beta propensity scale. Whatever scale I am showing is a beta propensity scale. Okay, higher the value beta, lower the value alpha. Emini surface accessibility scale. Similarly, we have another scale based on surface accessibility. Okay, so here you can see this kind of propensity scale. From this we can determine The surface, what for the surface is needed to know whether this antigen is binding with the, that is epitopic region of antigen is binding with the paratopic region of antibody. So we need the surface, isn't it? Even the B cell, top of the B cell receptor is a B cell recognition. Okay, so that's most important. The surface is important. And similarly, the third is the flexibility. Even the flexibility of amino acids can give an important parameter to determine the epitopic peptides. Okay, so based on the flexibility here also we can find a kind of a propensity scale. So through that we can determine which can uh, form the epitopes. The fourth is the Kolaskar and Toyenka. They have proposed based on antigenicity. The antigenicity is purely based on the physicochemical properties. So the physicochemical properties of amino acid sequence in the peptide. Okay, Kolaskar and Toyenka has come up with a kind of an antigenicity scale. And if you see the B cell epitope prediction softwares, we have plenty of tools in immunoinformatics. Other than IEDB, in IEDB we have this kind of a tool. We also have BabyPred. So this BabyPred is also present in IEDB, but this also present as an exclusive tool. Okay, in Raghava we have ABCPred LB2, and we also have private web servers for BCPred. So these are private servers. Whatever I am showing you here are the private servers. So we have plenty of tools. Hundreds of tools are available to predict the B cell epitope prediction. Okay, and in IEDB B cell epitope prediction, if you click the B cell epitope, we can find these kind of tools. The first tool is prediction of linear epitopes. So if you want to predict the linear epitopes, we have to click this. And if I want to predict epitopes from the discotope algorithm, we can use this, where it uses the solvent accessible surface area calculations. But whereas initially, whatever we predict linear epitopes, this is based on sequence characteristics. But here, the area, surface area characteristics. The third is the ELIPRO, epitope prediction based upon structural protrusion, that is based on solvent accessibility and flexibility. And we can also go with molecular docking and modeling of antibody structures using this URL. Okay, we also have few more tools related to antigen processing, presenting. I told you, no? So even if you see with respect to the structures of T cell receptors, B cell receptors, for modeling that T cell, B cell receptors, we can use this Lyra tool. So like this, whatever if you know the immunology, we can work with this kind of a tools and we can predict it. Now let me go ahead with the linear B cell epitope prediction. If I click the linear B cell epitope prediction, we get numerous algorithms. So we have algorithms like BabyPred 2.0, whatever present in default is 1.0, Chow Fastman, Emini surface accessibility, CarPlus, Kolosker, Parker hydrophilicity prediction. The only thing is that you have to either input Swissprot ID, which is an Uniprot ID, that is a 6-letter code, either input the 6-letter code or just paste the sequence alone in amino acid format. I mean, you should not paste the FASTA format, ignore the first line, paste the amino acid sequence alone and click submit. Either this and that is okay. So like I'm pasting the Uniprot ID. Okay, that is for Nipah W protein. And if you click submit, you will be getting the result or else paste the sequence. So after inputting the Uniprot ID, we'll be getting this kind of a result, a graphical result. Here, whatever peaks at the top or the best epitopes predicted. So if you see all the peaks, this is the best peak. This is the top peak. And next to that, you can find this peak. This is the second best peak. If this is first best epitope, this is second best epitope we have predicted. Third is this. Like this way, we can able to predict numerous epitopes. But if you have more than 10 epitopes and you want to screen just the best 10 epitopes, then I suggest you to increase the threshold. Earlier, the defaultly, the threshold fixed is 0.5. If you can able to keep the threshold up to here 0.6, check this. zero point 0.6, you can you are filtering few epitopes. Whatever present above the red line, that alone considered as epitopes. And whatever these we are eliminating, these peaks we are eliminating. You got it? So whatever peaks you find here, these we are eliminating, and we are and even we can fix with decimals 0.62, 0.64, so that even we can screen the best 10 epitopes or whatever of interest we are making. Okay, so not only we can see the prediction of epitopes based on the peak sizes, but also related to the sequence, that amino acid sequence residues, you can see here. So if you see this protein epivirus W, it comes around 450 amino acids. So all the 450 amino acid positions are mentioned here. Okay, from 0 to 450 amino acids are there, and these are the residues. And wherever you find the peaks, default I kept threshold as 0.5. So above 0.5, wherever you find the peak, that will be considered as epitopes accordingly. And earlier, whatever we find here from the 20th amino acid, see here, from 20th amino acid, you can see the peak is forming. The peak, whatever formed is here. So from 20 to this much, it comes around how much? 40, something like that. We find an epitope is formed. So 20 to 40, we can find E, E, E. Wherever you find E is an epitopic peptide formed. Wherever you find dots, no epitopes or screen based on the threshold. This is how you can find the results in graphical format as well as in this table format also. Okay. Now we can go to the next discotope prediction. In discotope prediction, these are trained on 75 X-ray structures of antibody protein complexes. Here we have the methods like Parker hydrophilicity, amino acid occurrence, number of contacts within 10 angstrom, and area of relative solvent accessibility. There are here also we have two types of algorithms. Discotope, the area under the curve is 0.71, but for discotope 2, the area under the curve is 0.73. A slight variation with respect to the algorithm. Whatever you find here, even here, you find the area under. Whatever you find area under, that gives the epitope. So here to here, whatever you find, is 20 amino acid to 42. 20 to 42 is what? One epitope, isn't it? Similarly, there also. Here in the discotope, remember, here instead of feeding the Nipah virus W protein sequence, that is uniprot ID, feed the PDB ID. So either you feed the PDB ID or upload it, and then you can click submit. Now let me go ahead with the comparison of B-cell epitope prediction methods. In the comparison of B-cell epitope prediction methods, I already discussed BPPRED, etc., isn't it? Now I'm showing you the Chow-Fasman. In the Chow-Fasman, we can see these kind of peptide peaks. So the same sequence, but whatever peak you find here, the amino acid residue positions are varying. It means that in each and every algorithm, so there we used a different algorithm. Here we used a different thing. This is based on beta turns, and based on surface accessibility, the best peak you got is around 20 amino acid. But there we got some different value. Here we are getting a different value. Here we are getting another different value. So you have to compare the algorithms, and whatever is shown more frequent, you can freeze that as the best. Like, however, I mentioned you in T-cell epitope, I've used 4 web servers, and from the most commonly occurring peptide epitope, I screened as the best one. Instead of filtering a best of one peptide epitope, you can filter around 20 epitopes in each and every algorithm and check the matching kind of a peptide. This is how we can fix the workflow. Now let me go ahead with the next Parker hydrophilicity. Here also we have a different kind of a peaks. Okay, the top 2 peaks alone I am showing, we find in different regions for the same protein sequence. Because flexibility is different, hydrophilicity is different, surface accessibility is different, beta turns is different. So obviously we find different kind of epitopes. That's the reason. Now, whatever epitopes we predict, finally we have to dock with a protein, isn't it? Whatever peptides, however, we have predicted in T cell epitope, we docked with HLA molecule. How about here? Here in B cell epitope, we have to dock with pathogen recognition receptors, PRR, refer to the pathogen recognition receptors. Some 3 years before, I believe, even those who worked on pathogen recognition receptor received the Nobel Prize. Whatever I'm explaining in the vaccine design, for the from 2014 to present year, we can find around 4 Nobel Prizes are awarded in different sectors. Okay, so pathogen recognition receptor, one of the pathogen recognition receptor is a toll-like receptor. In toll-like receptor, you can find for bacteria, like for example, whatever B-cell epitope we are designing, if we are targeting the bacteria, the bacterial membrane, we have lipopolysaccharide, isn't it? So we have toll-like receptor 4 in our body, so which is going to combat, which is going to how you find the traffic police can have the barrier gate near the end of each and every region. Similarly, if bacteria is coming inside our body, then obviously this TLR4 can object to come inside. Okay, so this is the major target receptor for the bacteria. And if you see this double-stranded RNA kind of a viruses, TLR3, and single-stranded RNA, TLR7 and TLR8. So these are the proteins we talk with our peptides, whatever screen peptides. Okay, and now let me take you to the CABS talk. CABS dock, this is another tool. Here in this CABS dock tool, we can dock a peptide with a protein. For this, we need a peptide sequence and protein. Like whatever sequences we have screened from a linear epitope prediction. So that peptide epitope sequence we can take, and we can take this toll-like receptor, and then we can dock. For this, we can use CABS dock. URL is mentioned below. Similarly, HADOC. In HADOC, we can dock protein-protein. Okay, HADOC also called as high ambiguity driven protein-protein docking. Okay, so you can use this URL, and if you have an antigen and antibody, like earlier I mentioned, hemagglutinin antibody, human antibody, that two molecules, whatever you have in PDB formats, you can feed as an input, and we can find the results. This is another server for protein-protein docking and also peptide-protein docking. We can also try with the ligand docking. We have plenty of tools actually. But in our case, if you dock peptide with the protein, we can use FlexPepDock. FlexPepDock. So in Rosetta Online server, this is the website URL. So if you go to this URL, if you click this FlexPepDock, you have to feed the peptide sequence as well as peptide structure as well as protein structure. Okay, then obviously you can able to see the docking. Not only you can try with this kind of FlexPepDock for the vaccine design, we need even if you are designing the vaccine using RNA, we can also work with this. Symmetric docking is available. Beta peptide design is available. So many tools are available here. Rosetta covalent peptide dock. Okay, so here in this protocol, you can use this submit, and you can upload the uh, peptide and protein for docking. And now let me go ahead with another tool called Violin. This Violin is a web-based vaccine database for veterinary ends. Like if you want to develop vaccines, in the earlier classes, in the earlier internship courses, even veterinarians have joined. Doctors, MBBS, MD doctors also have joined. They find these kind of explanation is also useful for them. Even if you're working any projects in Tanvas or any other veterinary research institutes, of course, you need these kind of website database. Till now, whatever we're discussing is up to the humans, isn't it? This is specifically to the other animals, not only domestic animals, even we have vaccines for wild animals. Okay, violin. Plenty of tools are available. Now, let me go ahead with these few tools in Violinate. The first is we also have for human, of course, human vaccination, HUVax, licensed human vaccines. I don't know how many know that how many vaccines we have taken from birth. At this age, what vaccines to be taken? So some we should have this kind of a knowledge, isn't it? So if you want to know what vaccines are available in the in India, in the world, so we can also use these kind of database actually. So only thing is that you have to select a pathogen, okay, and then if you select for search, you will be getting the list of vaccines. If you specifically want a kind of a vaccine, the pathogens type the keyword and you can search for it. Similarly for animals other than human, VVAX, like HUVAX, we have VVAX. So you have to type select a pathogen and search. No need to type anything also. We have plenty of information generated from that you can select specific thing. Protection, protective antigens. Okay. DNA vaccine, DNA Vax TV. So whatever we have taken, Covishield is a form of a DNA vaccine, isn't it? So of course, SARS-CoV-2 is a zoonotic pathogen. The viruses came from the animals, isn't it? So, of course, there is a relation between whatever vaccines for the human may also work for the close related animal, close related animals of human. Okay, but if you want to go specifically for treating SARS-CoV-2 for dogs, that's a major pet animals, isn't it? Next to that, feline cat. We can also design vaccines specifically for that. First of all, before designing, we can check whether the vaccines are available or not. Okay, using this. So DNA vector we have completed. Next we come across VaxVec, a database of recombinant vaccine vectors and recombinant vector vaccines. Okay, so specifically for recombinant vector, we can, so whatever we have discussed as Covishield is of course a vector. They have used a recombinant technology, and of course that is also linked with this kind of VaxVec. That Covishield also linked with this. Firmugen, a database of virulent genes. We have types of major two types of vaccines: live attenuated vaccine, killed vaccine. Okay, so this live attenuated vaccine, where we remove the virulent genes and use that kind of viruses to kill uh, the live viruses, isn't it? So for this also we have an exclusive database called Firmugen DB, VAXA, Vaccine Animal Response. See, numerous animals are mentioned here: mouse, human, chicken, monkey, macaque, monkey, guinea pig, cattle, rabbit, hamster, rat, fish, cat, baboon, horse, ducks, dog, sheep. For all these animals, we can able to get the vaccines available. Not only with respect to the viruses, with respect to the cancer, with respect to the autoimmune diseases, what vaccines are available and what is the mechanism and uh, who is who the publications of the vaccines, who has discovered the vaccines. All this information we can get from this vaxar, vaxism, vaccine mechanisms. Okay, different vaccine mechanisms you see. But you see, we don't have data for everything. Okay, for human also we have only one. But for mouse, we have plenty of vaccines. Okay, so we can able to check whatever is available, that alone we can able to see. We can't find for everything, but whatever is available we can able to find the information. Vaxism, vaccine mechanisms. So like how the vaccine for African swine fever can work. Okay, for the African swine fever virus, all this information you can see: introduction, pathogenesis, animal models, protective immunity. If you're working on a design of vaccine, obviously we should check first initially with which is the best animal model. I told you that whatever I worked with Nipah virus W protein in immunoinformatics, whatever I published. So this paper helped a PhD scholar to take a research to initiate and to complete the in vitro studies on this kind of virus. Okay, in a vaccine research institute. So like this, you can find some information earlier before proceeding on to the research. Okay, even you can use this website. And now let me go ahead with the overall kind of a computational vaccinology. So till now, whatever we discussed is something related to peptide-based vaccine design, isn't it? But we also have other types of vaccines. Like just now I explained you recombinant DNA vector vaccines, mRNA vaccines. So many things are available, isn't it? Other than the peptide. So how these vaccines are designed, how this rational vaccine design is carried out. Okay, so this will be discussing in this slide. So if you see here, the types of vaccine other than epitope, if you consider the epitopic based vaccines are used for construction of multitopes. The best multitope vaccine I explained you is the COVAX. The initial strain of SARS-CoV-2, you can find around 99% efficacy. I told you, when compared to Covishield and Covaxin, this is the best vaccine identified at that time, and this was present only in US. Okay, so we don't know about this. And the next vaccine, what is, next computational technique for the vaccine, which is most important, is the modeling tools. Homology modeling, we can use as exclusive tools for going with homology modeling, other than Swiss modeler. For the vaccine antigens, we can go with this homology modeling tools present in that vaccine databases and tools. Then number three is the antibodyomics. See, the antibody structure is something different. You can't take the horse, just a Swiss modeler to model the antibodies. We need a specific tools. For the antibodyomics, I suggested to go with Discovery Studio antibody modeling. Discovery Studio antibody modeling. Schrödinger also, we have a separate module for antibody modeling. Modeler is different. We also have Discovery Studio Modeler. This is used for modeling any type of proteins. But for the specifically modeling the antibody, what is the special feature in the antibody? If you see the antibody, we have a light chain fragments and the heavy chain fragments, isn't it? And the surface here you can find, this is most important for antigen binding. Here in the surface, we can find so many fragments, SCFE fragments. Even a part of a sequence is most important for binding. And in some proteins, in some antibodies, you see, it can bind with the antigen 1. One arm is going to bind with antigen 1, and another arm is going to bind with another antigen. Okay, antigen 1 and antigen 2. So this concept is called avidity. And if both the arms is binding with the same kind of an antigen A1, then it is affinity. Whether our antibody is having an affinity towards the antigen or not, or it is having an avidity, can target multiple diseases. For example, MMR, measles, mumps, rubella vaccine. A single vaccine is targeting 3 different viruses. If so, even we have these kind of a specificity. Okay, got it? Multi-targeting things. So there is a lot to know even in this antibody engineering. Like how we discuss drug engineering. In drug, we can use a fragment-based screening. Like similarly, even in antibody, we can go with a different antibody engineering technique. So more about the antibody modeling, we have an exclusive commercial tools to build, to precisely develop the antibody structure. We can use these tools. And number 4 is the codon optimization. For some viruses, we can inhibit the translational step. DNA is going to transcribe to RNA, that is mRNA, and RNA is going to translate to protein. So either we can block this step, RNA which code for a protein, or we can block this step. Okay, so if you can able to block this even transcription kind of steps, okay, so of course we can go with this kind of a codon optimization tools. So like even in drug mechanism, you can find, isn't it, these kind of transcription inhibitors, translational inhibitors, Similarly, here we can use the codon optimization for the prevention of the viral infections or bacterial or any pathogen infections. Now let me go ahead with the next thing is scaffolding tools. Scaffolds, like how a drug can interact, drug can link with another drug in the form of a prodrug. So even, if only if a drug is released and if it is reached to a desired target, then only a part of a drug is going to eliminated, and earlier the inactive drug, whatever we have, is going to convert as active drug. Like how if a satellite is released from the rocket, similarly in a drug, if you have two parts, and if you take this two part of a drug, what we call it as a prodrug, that is inactive drug along with the carrier. If the carrier is released, the part whatever we find active drug can give the desired biological effect. Similarly, here also we have scaffolds like RNA scaffolds. Okay, that co-vaccine, how it works. Okay, so RNA vaccines. For this, we need this kind of a scaffolding tools. Okay, we have plenty of computational techniques for the vaccine design. Now let me go ahead with the patch talk results. Actually, for protein talking, I didn't show you any kind of results, isn't it? I'm showing you with the patch talk initially. In patch talk, initially, whatever I showed, hemagglutinin, this is an antigen. The hemagglutinin antigen is larger in size when compared to human antibody. So what I did is, I have taken this as a macromolecule. That is, a viral hemagglutinin antigen is a macromolecule now, and human antibody is a small molecule when compared to that. But both are proteins, and both we have a PDB files. So I uploaded it and I use the email ID. So I can able to get the scores. I am getting different conformers. Protein is talking with another protein with different conformers. We are getting different complex files, result 1, result 2. The best, of course, is the first one. So it is sorted according to the ascending order. So you can try with one of the top three also. Okay, like whatever we have result one.pdb, you can give it as an input. Okay, even that also has to be fed as an input along with the receptor and ligand for FireDoc2. That is, the output of PatchDoc can also serve as an input for FireDoc. So other than giving an antibody and a hemagglutinin, you can there is also one more file which we have to feed, that is a result1.pdb. That after feeding all these three files, I can able to get even this energy. So this is what we can report. That is global energy I got is minus 22.19. Okay, so we can find different conformers, and these are the results. Other than global energy and attractive and reverse repulsive, so these information like protein talking, we have multiple tools: patchdoc, hexdoc, firedoc. So patchdoc, firedoc, we can use like this. Now let me go ahead with something about vaccination. So till now we discussed about B-cell epitope prediction, veterinary vaccines other than human vaccine database, Violinate. Then we discuss something on peptide protein talking tools, protein talking tools, and how about the outputs, how to infer the outputs, how to take the inputs. Then only if we know how to take the inputs, then only we can feed the sample files and get the output. So all these things we have discussed up to present. Now let me go ahead with this slide about how to take the vaccine and when to take the vaccine. So I'm giving with few examples. The first example is BCG, Bacillus calmette-guerin. If you take at 8 a.m. early morning, you see high cytokine secretion. I told you this is the most important immune protein, interferon gamma, to combat viruses. TNF alpha, tumor necrosis factor alpha, most important to combat cancer. And related to inflammation, we have this IL-1 beta. Now, if you see these high cytokines are secreted in the early morning if we take BCG. But at 6 p.m., very less cytokine secretion. So no trained immunity. So also which duration we take, that also matters. And number two, influenza, 9 to 11 a.m. Higher antibody, humoral response. Here we seek the humoral response in influenza. And even in Nipah virus, when compared to cellular immunity, humoral immunity is the best one. I have published T cell epitope prediction, but when compared to T cell epitope prediction, B cell epitope prediction is more significant. That might I have tried in another publication. I have yet to do that. Okay, I just doing some other research, and just giving an example. For few viruses, humoral immunity is most significant when compared to cellular. So these cytokines are released because of cellular immunity. If cell interacts, these cytokines are released. So that is also important, not just when you are taking this. Okay, I think you find this a significant slide. Now, if you see at the evening 3 to 5, lower antibody response. Okay, so what you can understand, even the metabolism is also plays a very important role. So only on the morning and evening, you find the difference in secretion of antibodies or the cytokines from the cells. Still, even in the SARS-CoV-2, if you have taken at 9 to 11 a.m., it's very active. You will be getting more antibody. You will be getting more B and T lymphocytes. That is, memory cells are created. But if you take at evening, so everything is gradually decreased. Antibody modeling, squad. So in in Tamil Nadu, in my regions, so the vaccines are given only up to 9 to 1 p.m. I believe. Everyone they closed. I don't know at that time. Later, I just find these kind of an information. If you have taken after 3, obviously everything got reduced. Humoral immunity, cellular immunity. Now let me go ahead with the antibody modeling. I told you if you want to work specifically, then I told you to go with the commercial softwares. Once it's Schrodinger. So in Schrodinger for antibody design, we have an exclusive program. Okay, so we should also have knowledge on antibody engineering related to that. And we also have Discovery Studio. Other than Discovery Studio Schrodinger, we also have DNA Star. This is also again a commercial software. Only the commercial software, so we can able to develop a novel antibodies. Whatever what is that in Bangalore, what is the Biocon? Of course, they use initially computational techniques to come up with antibodies, and then they synthesize this kind of an antibodies. Okay, so this is how we can able to understand this thing. So related to the B cell epitope prediction, we have very less time. Just give me some 4 minutes to complete. So first go to the IEDB. So go to the B cell epitope prediction. If you click this B cell epitope prediction, you get all the tools related to the B cell epitope prediction. If you want to go specifically to the linear, even we can click the linear from inside. We also have tools for antigen sequence, Discotope, ELIPRO, different tools. Can I go with Discotope? Meanwhile, sir, okay. Now I got this B-cell epitope prediction tools. Plenty of tools are available. For linear epitope prediction, click this. After this, what you have to feed? Uniprot ID. So either Uniprot ID or Nipavirus W protein sequence. Can we check what Uniprot ID we have given? P0C1C7. P0C1 P0C1C7. Okay, so this is the ID, Swiss Prot ID. Default I kept like Webpred 2.0. Click submit. So we'll be getting the results. However, I showed you in the PPT. Later, we can increase or decrease the threshold based on how many epitopes we want. So we can screen the epitopes based on 10 to 20 epitopes, and we can compare with other algorithms to find the most frequently observed epitope. Okay, this is how we can filter as the best epitope. Later, we can use the peptide protein docking servers. Before proceeding to that, we should also, if there is a need to predict the structure of peptide, we can also predict the peptide through PepFold3 server. To predict the PepFold3, so we can use this PepFold3. So to predict the protein peptide structure, go to the pepfold 3 and then run pepfold 3. Then here you have to type the peptide sequence. If you feed the peptide sequence and if you click run, obviously we'll be getting the structure. So it takes just 1 to 2 minutes. Within 2 minutes, we'll be generating the structure. So antibody is predicting, isn't it? The server is working, so you can find. We're working in online tool, not in offline tool. So based on the frequency of internet, we can get the result. Of course, I'm working in i7 and i9 laptops. This itself, and of course, I am also using internet frequency. Go to internet frequency. Let me see. But I'll be showing the result. Any doubts you can ask me now. Almost I have completed the session. Only thing is that whatever I pasted the URLs in the WhatsApp, you have to click inside and you search it in random and you can work on it. So for example, violineta.org. So this is what I told as a veterinary vaccine and human vaccine database, violin. So go to this violin. So after that, you can, if you want to search specifically vaccine vectors, anything, you select anything. For example, VVAX initially, veterinary vaccines. So any animal specifically, baboon boards, you select anything. Or if you want to select any specific kind of a virus, like Bacillus anthracis, last year, Bacillus anthracis anthrax got infected uh, 3 deeds in IIT Madras. When I'm working in IIT Madras, I found last year 5, how many? For nearly 4 to 5 deeds or dead just because of this. So just selecting this Bacillus anthracis searched, or if you want to specifically search an organism, you can search it. Licensed vaccine. So you can search for the specific thing. But they have done very stringent protocols to eliminate the contaminated, that animals. Okay. This is something related to this Bacillus anthracis. What are the types of vaccines? So we have to go inside and find this information. Okay, run pepfold 3 and Discoto tools. What happened? The server got hanged. Still I'm running. But however I got the results. Similarly, you can get the results here. For PDB-ID, I'm just giving an example, whatever they have. But it's a must to give even the chain ID. Other than giving the PDB-ID of a Nipah virus W protein, this is not a Nipah virus W. I'm giving an example. If you know the PDB-ID, you can feed the PDB-ID or just upload the model structure, whatever we have, and then enter the chain. Entering the chain name is also mandatory. If you click this, here you need the structure of the pathogen protein, then only it can predict the peptides. Okay, I can close few things so that you can see the results. So we are getting this result. 3D view, table view is there. So this is from Discoto. Application is getting loaded. So this is something about the results. The table view is also there. You can see Discotope score, propensity score. So this is how Discotope result looks like. And the B-cell epitope, I think it's the server is slow. You can check. Okay, if you have any doubts, you can ask me after practicing. Okay, any doubts you can ask me. I have completed today's session, and on tomorrow we'll be discussing on machine learning and artificial intelligence. Till now we have discussed on drug and vaccine design. So only from tomorrow onwards we discuss the applications of AI and ML. Okay, and of course the last session is on Python programming. Okay. Any doubts I can ask me at present. If no doubts, thank you very much for your patience and interaction. Have a great day to you all. And tomorrow and day after tomorrow, we have the sessions. And as I mentioned, after completing the last session on Sunday, we can have some 4 days time, 4 to 5 days time. I can give 4 days time to complete the MCQ. The MCQ, whatever I said, is very easier to attempt. So only the basic things you should know. So I didn't ask any twisted kind of an MCQ. So it is easier to attend. So best wishes to you all. Okay. Have a great day. Bye.